Hello, everyone. I'm Ron Vale, and I'd like to welcome everyone to another week of life science across the globe. We're now in our second round of talks, and we finished two talks from North America, and now we travel down south to our friends in South America. And I'd like to welcome back the University of Buenos Aires and the National Research Council, or also called CONICET. And now I'd like to introduce Gabrielle Rabinovich, who is the Senior Investigator of CONICET and Professor of Immunology at the University of Buenos Aires. Gabrielle has done pioneering work in the field of glycobiology, particularly as related to the fields of immunology and cancer biology. And on the basis of his many research accomplishments, Gabrielle was elected as a foreign member of the U.S. National Academy of Sciences. So thank you very much for joining us, Gabrielle, and please go ahead and introduce today's speakers.
Thank you very much, Ron, for this introduction. It is my great pleasure today to introduce the two speakers, Belén Elgoyhen, who will deliver the science talk, and Mario Peceni, who will deliver the culture talk. Belén Elgoyhen graduated as a biochemist from the University of Buenos Aires and obtained a PhD in biochemistry from the same university. She then moved to the USA for a postdoc at the Salk Institute under the direction of Dirk Heinemann, where she cloned novel subunits of the family of nicotinic acetylcholine receptors, which are expressed in the inner ear. Since then, Belén has made groundbreaking discoveries in the field of auditory physiology. She has gone all the way from hardcore basic science to the search for treatment of inner ear diseases. Belén is full professor at the Department of Pharmacology, University of Buenos Aires School of Medicine, and vice director of the Institute of Molecular Biology and Genetic Engineering of CONICET. She has been recognized with several awards. These include the International Research Scholar of the Howard Hughes Medical Institute, Guggenheim Fellow, and member of the Latin America Academy of Science. She was also awarded with the Investigator of the Nation Award granted by the President of Argentina, the Third World Academy of Science Prize in Biological Sciences, and the L'Oréal UNESCO Award for Women in Science in Latin America. The title of her talk today will be The Brain Talks Back to the Ear, the Efferent olivocochlear system.
Good morning, good afternoon, or good evening. It is a great pleasure to be part of this Life Sciences Across the Globe seminar series, and I want to thank Ron for the invitation. My lab has worked over the years in the physiology and genetics of hearing. And during this half an hour or so, I'm going to show you some of the results we've obtained. It is within the inner ear where the sensory epithelia for hearing lies, and this is known as the organ of Corti in mammals. The organ of Corti has different types of cells, but for the sake of this talk, we're just going to concentrate in this purple type cells, which are sensory cells. And in mammals, these are divided into inner hair cells on the one hand, which are the primary phonoreceptor cells in charge of transducing auditory signals into electrical signals that can then be sent to the central nervous system through the auditory nerve. But mammalians also have a different type of hair cells known as outer hair cells. And these outer hair cells are in charge of amplification of the incoming signal right at the periphery. And this is brought about by a novel mechanisms, which is a mammalian innovation known as somatic electromotility, whereby changes in length in the outer hair cells, that is elongation and shortening, elongation or shortening, are produced based on changes in the membrane potential, resting membrane potential of the cell. And these changes in length produce amplification and movements within the auditory epithelia. And this leads to amplification. Now, this somatic electromotility not only produces amplification, but it also fine-tunes the basilar membrane of the organ of Cordy, whereby now each point along the length of the basilar membrane is most sensitive to one particular frequency, high frequencies at the base, low frequencies at the apex. And this fine-tuning or tonotopic organization is in part due to this somatic electromotility of the outer hair cells. Now, if we zoom into the organ of Cordy and look at the innervation of hair cells, we see that hair cells have two types of neurons contacting them. On the one hand, these red neurons, which are afferent neurons, bringing information from the periphery to the central nervous system. But the hair cells also receive a feedback information from the central nervous system directly to the base of the outer hair cells, and this is known as the efferent system. And we're going to concentrate in this MOC green system, which is medial olivocochlear efferents, which make contact at the base of the outer hair cells, release neurotransmitter, and inhibit electromotility, inhibit somatic electromotility via hyperpolarization of these outer hair cells, and therefore inhibits amplification. If we zoom in and look at the neurotransmitters which are released at the synapses, we see that afferent synapses are glutamatergic, whereas medial olivocochlear efferent synapses are cholinergic. Acetylcholine is the neurotransmitter released at that synapse. And I would like to already from the start make the point that although it is only in mammals where medial olivocochlear efferents have to modulate or inhibit somatic electromotility, efferent system exists in all hair cells of all auditory papillas of vertebrates. However, it has specialized in mammals in order to modify or modulate or inhibit somatic electromotility. Now, quite a long time ago, probably over 20 years now, we were able to decipher the molecular nature, the molecular structure of the cholinergic receptor present at the efferent outer hair cell synapse. And we now know that it is a ligand-gated ion channel, gated by acetylcholine, and this ligand-gated ion channel belongs to the nicotinic family of cholinergic subunits, and it's a pentameric assembly of two subunits, alpha 9 and alpha 10, in 2 alpha 9 and 3 alpha 10 stoichiometry. Now, over the years, we not only deciphered the molecular nature of the nicotinic receptor, but we and others were able to discover the different mechanisms and molecular actuators at this synapse. And we now know that when acetylcholine is released, it binds to the binding side of the alpha 9, alpha 10 receptor. This receptor is highly calcium permeable. Calcium goes in when the channel pore opens, and this calcium activates a nearby SK2 potassium channel, and potassium goes out of the cell. And this leads to hyperpolarization of the outer hair cell and inhibition of electromotility. Now, this is seen when if we now do electrophysiological recordings, patch clamp recordings from outer hair cells. We can see that currents, we see changes in current when the medial olivocochlear efferents are stimulated, and this is seen first through an inward current, and this inward current is due to channel ions fluxing through the alpha 9, alpha 10 receptor. But this influx current is immediately overridden by a long-lasting and bigger outward current, which is due to the SK2 activation of SK2 channels and efflux of potassium. And I would like to mention, before I forget at the end of the talk, that most of the physiology in the lab was started by Eleonora Katz in collaboration with Paul Hughes and Elizabeth Glowatsky in a collaboration with them at Johns Hopkins. Now, one intriguing question of efferent and afferent synapses at the base of these outer hair cells is how this high calcium entry through this efferent mechanism produce hyperpolarization and thus inhibition, instead of facilitating glutamate released to afferent fibers and thus excitation, given the fact that these efferent and afferent fibers are in very close proximity at the base of the outer hair cells. So Marcelo Mogli and Juan Gutman were able to do a series of experiments whereby they measured calcium imaging coupled to electrophysiological recordings, both from hair cells and afferent recordings, in order to study the mechanisms underlying distinct excitatory and inhibitory calcium roles within the small compact hair cells. And they were able to describe how an unusually high intracellular calcium buffering coupled to an organelle which is present at the base of these outer hair cells, which is known as subsynaptic cistern. Both of these mechanisms provide efficient compartmentalization and tight control of calcium cholinergic signals, and thus synaptic efferent calcium spillover to afferent synapse does not take place, and efferent synapses are always inhibitory. So with the cloning of alpha-9 plus alpha-10, we were able to finish and describing the landscape of nicotinic acetylcholine receptor subunits. And this family of subunits is now divided into the muscle type receptor subunits, which assemble to form the receptor at the neuromuscular junction that produces contraction of skeletal muscle. Then we have a plethora of neuronal subunits, which assemble in different combinatorial assemblies to form different receptors that are present in the central nervous system. And finally, we have the alpha-9 and the alpha-10 subunits, which are the only ones expressed in adult hair cells. And moreover, alpha-9 and alpha-10 have a very restricted expression pattern. For example, they are not expressed in the central nervous system. Now, from early on, from the cloning of these two subunits, we realized that although they belong to this family of nicotinic acetylcholine receptor subunits, they are distant members of the family because the sequence identity at the protein level between these two subunits and any of the other member subunits is very low. And moreover, the pharmacological and biophysical properties of these receptors, the receptor assembled from alpha-9 and alpha-10, compared to any other nicotinic ACH receptors, is quite distinct. For example, nicotine, which is the canonical agonist that, that characterizes this family, is not an agonist of alpha-9 and alpha-10. So very early on, we realized that there was something quite distinct going on in the evolutionary history of these two subunits. And there were Lucia Francini, Marcela Lipovsek, and Irina Markovic got interested in the molecular evolution of the entire family of nicotinic ACH receptor subunits, and they compared the evolution of all other neuronal or muscle subunits to that of alpha-9 and alpha-10. And using different in silico molecular evolutionary analysis, they were able to reach to some interesting conclusions. For example, they performed a phylogenetic tree, and in this phylogenetic tree, they included almost 400 sequences from the 17 different nicotinic subunits, belonging to 29 vertebrate species. And we can see that the family is divided into different clades. On the one hand, the alpha-9, the alpha subunits. On the other hand, the non-alpha subunits. Then we have a different clade which includes the alpha-7-like subunits. And finally, we have a different clade known as the alpha-9-like subunits. Now, if we zoom in to this clade, we can see a very particular subgrouping with alpha-10 subunits unique in presenting a segregated grouping of orthologs with non-mammalians alpha-10 subunits as a sister group of alpha-9 and mammalian alpha-10 subunits as an outgroup to this alpha-9 non-mammalian alpha-10 subunits. And using different molecular evolutionary techniques, Lucia and also Marcela and Irina were able to determine that alpha-10, mammalian alpha-10 subunits acquired non-synonymous substitutions at the coding region of the protein in mammals, by which now mammalian, non-mammalian alpha-10 subunits are quite close in sequence identity to all alpha-9 subunits, whereas mammalian alpha-10 subunits diverged in sequence, in amino acid sequences. Now, if a protein which has very important domains that need to be conserved in order for that protein, in this case, to work as a channel, has acquired non-synonymous substitutions in the coding region of that protein. And these non-synonymous substitutions have been fixed during evolution. This probably means that this protein has acquired novel functions or novel functional properties. So Irina wanted to test this hypothesis, and what she did was to compare the functional properties of different receptors, neuronal receptors, for example, a heteromeric receptor, alpha-4 beta-2, another neuronal receptor, a homomeric receptor, alpha-7, and an alpha-9, alpha-10 receptor across different species. And she chose frog, chicken, and rat. And she compared the functional properties of those receptors. She cloned those subunits, heterologically expressed them in a heterologous system, and compared the properties of different functional properties of those receptors. For example, agonist potency, the desensitization kinetics, calcium modulation, permeability, etc. for each one of these receptors from these different species. And she could see, although this is a very complex table, it's easy to interpret. She could see that across species, all properties were similar in the case of neuronal receptors, alpha-4, beta-2, and alpha-7. For example, if we look at desensitization kinetics, it has a score of 70, a score of 70. And this score of 70 is the same for frog receptors, chick receptors, and rat receptors in the case of this neuronal subunit and this neuronal receptor. And this is the case for each one of these functional properties for these receptors that she analyzed. Now, if we look at the alpha-7 receptors as cross species, we see that this desensitization has a score of around 2, 3 for all receptors. So this property is the same across species. However, if we now go and look at the functional properties across species of alpha-9 and alpha-10 receptors, we see that this score changes across species. We go from 14 to 64. And this is true for any one of the functional properties we analyzed for the receptor. So as a conclusion, we can see that while all members of the nicotinic ACA receptor subunit were conserved and were under purifying selection during evolution, alpha-9 and alpha-10 receptors were under positive selection to acquire non-synonymous substitutions at the coding region, and this led to changes in functional properties. And these changes in functional properties most likely were correlated with the acquisition of a novel function in mammals, and this novel function is likely to inhibit somatic electromotility, which is a mammalian novelty. Now, Lucia Francini went further in the molecular evolutionary analysis, and she looked at other proteins which are involved in somatic electromotility in outer hair cells. And we know that somatic electromotility is based on a very rich and complex extracellular matrix in the lateral wall of hair cells. And Lucia found that spectrin, for example, spectrin has also been under positive selection, which is part of this extracellular matrix. And moreover, prestin, which is a molecule which is highly packed in the lateral plasma membrane of the hair cell, and which is the motor protein involved in which is the basis of somatic electromotility, has also been under positive selection in mammals. And moreover, in a recent high throughput analysis of molecular evolution of 1,300 genes, Lucia Francini and a student in her lab, Francisco Picciotano, found that 13% of these genes have been under positive selection. So it seems that hair cell genes are a hotspot for positive selection as the acquisition of novelty and innovation during evolution. So far I have told you that the medial olivocochlear system is an inhibitory system, and it is a cholinergic receptor. The cholinergic receptor present there is a nicotinic type receptor formed by alpha-9 and alpha-10 subunits with a 2 alpha-9, 3 alpha-10 subunits stoichiometry. This receptor is highly permeable. It is a distant member of the family, and being a distant member of the family is based on the fact that it underwent a distinct evolutionary trajectory. Now, during the last minutes, I would like to shift gears and talk about a little bit about the physiology of the efferent system. And we know that the function of the efferent system is important for several aspects, or has been postulated to be important for several aspects. We know it is a gain control system. It inhibits somatic electromotility. It has been proposed that it mediates selective attention, for example, visual versus auditory. For example, when it diminishes auditory input to the central nervous system, when we need to concentrate on, for example, in a visual task, like reading. It controls masking from background noise. It shapes the normal cochlear development. And finally, it protects the inner ear from injury produced by over loud sounds. And I'm going to just concentrate on this last point, which I mentioned here. Now, the exposure to loud sound is one of the most important hazards that we are exposed nowadays in our busy cities. And exposure to loud sounds produce damage to the inner ear, and this is translated into two things. On the one hand, it produces hearing loss, and on the other hand, it produces tinnitus. And this is based on the fact that loud sounds produces first a disarrangement of the cilia in the apex of the hair cells, but eventually it leads to loss of hair cells, both inner and outer hair cells. If we look at a normal organ of Cori epithelium with 1 row of inner hair cells and 3 rows of outer hair cells, after noise, we see damage and loss of hair cells. And this loss of hair cells is irreversible, since these hair cells cannot regenerate. And as I said before, this leads to hearing impairment. If we look at an audiogram, we lose sensitivities to some frequencies, but it also produces tinnitus, and tinnitus is a phantom sound perception, which the people which have tinnitus hear the sound in the absence of an external sound source, and this is very debilitating for most of the people. And this one of the main known causes leading to tinnitus is the exposure to loud sound. So in order to study the function of the efferent system in all these different things that I numbered before, over the years we generated different genetically modified mice. And I'm just going to show you results from 2 of these mice. On the one hand, we generated an alpha-9 knockout mouse. And in an alpha-9 knockout mice, the efferent system does not work. Even given the fact that alpha-10 is still present, the efferent system, the mice are completely de-efferented, and this is seen here. If we do patch clamp recordings from hair cells and we apply acetylcholine, we don't see any ACH responses to the agonist. So these mice are completely de-efferented. But we also generated a different type of mice, which has a gain of function mutation within the alpha-9, alpha-10 receptor. And we did this by introducing a point mutation in the pore of the channel. And this point mutation leads to an increased gating kinetics of this receptor. And this increased gating kinetics leads to an increase in the strength of the efferent inhibition. So if we look at, for example, the postsynaptic currents recording in this knocking mice, and what we're doing here is we are stimulated the medial olivocochlear efferents and recording from outer hair cells, we can see postsynaptic currents, which in black are the wild type ones, and in red the ones from the knocking mice. We see that currents in knocking mice are really prolonged in time. And now if we look at the hyperpolarization produced by efferent stimulation, we see that in wild type mice, we see this intermittent hyperpolarization of the outer hairs of the hair cells. But in the knocking mice, we see a prolonged and big and sustained hyperpolarization of these hair cells, which leads to a sustained inhibition of somatic electromotility and a sustained effect of the medial olivocochlear system. And we have done different things with the genetically modified mice, but I'm just going to show you just one result that we have obtained lately, and this is the work from Luis Boero and Maria Eugenia Gomez Casati, where they traumatized mice, both wild type knockout and knocking mice, by exposing them to a very loud sound during a period of time. And then they analyzed auditory thresholds to see if these mice heard better or worse. No? And they did this by recording auditory brainstem responses. So an increasing threshold in ABRs means that the animals hear less, and they recorded ABR thresholds at different frequencies, both in wild type, knockout, and knocking, and the control conditions in black 1 day after exposing the animal to overly loud sound, and then 7 days after the exposure. And you can see that this is the ABR thresholds in wild type. And when these wild type animals are traumatized, here in red, thresholds go up, but this is reversible and it comes back to normal 7 days after exposure. If we now look at the knockout animals, which do not have an efferent system control, after traumatizing the animals, thresholds go up, animals hear less, but this is irreversible. These thresholds do not go down to normal. So therefore, this means that these animals, knockout alpha-9 animals, are more sensitive to this noise-induced hearing loss. And now if we look at the knockings, which have an increased efferent system, we see that these animals are not even traumatized. They don't have an increase in threshold. So these results show that the extent of the damage to the inner ear produced by the exposure to over loud sound is inversely correlated to the strength of the medial olivocochlear system. The less effect of the medial olivocochlear system, more damage to the inner ear, and vice versa. And in a very recent work that I'm not going to show you today, again, Lucho Boero, Valeria Castaña, and Maria Eugenia Gomez Casati have found that in this alpha-9 knocking mouse, and now looking at aged mice, they have seen that this alpha-9 knocking mice have less decrease in hearing abilities with aging. So these animals, alpha-9 knocking mice, are prevented from age-related hearing loss. So these are very interesting experiments, and this suggests that probably strengthening the activity of the efferent system can be used as a therapeutic approach in order to prevent damage to the inner ear. And we are now starting to look for compounds, small compounds, that act as allosteric modulators of this alpha-9, alpha-10 receptor, cholinergic receptor present at the synapse between the medial olivocochlear efferents and outer hair cells, with the hope of finding some compounds that can be used in therapeutics to the treatment of hearing loss. So in summary, over my talk, I've shown you that it is alpha-9, alpha-10 nicotinic receptors, the one that mediate the synapse between efferent fibers and outer hair cells, that these receptors have a very strange or different molecular evolution compared to other members of the family, and that this medial olivocochlear system protects the inner ear from acoustic injury. Before I finish, I would like to acknowledge external collaborators. I mentioned Paul Hughes from Johns Hopkins, but also Stefan Messon and Charlie Lieberman from Harvard Medical Institute. Of course, all funding agencies from Argentina, but also from abroad. And in special, I want to give a special thank in this occasion to the Howard Hughes Medical Institute International Program for Latin America and Canada, that over 15 years when the program existed, funded this work. And finally, of course, all the people who made all this possible. These are the faces of all of the students, postdocs, and undergrads that have gone through the lab and have made all these wonderful works. Some of their names I mentioned over the talk, but I don't have time to acknowledge all of them now, but I would like to, of course, thank them all. And thank you. And I'm open for questions.
Thank you, Belén. That was a very interesting talk. Let's jump right into some of the questions. First, you mentioned that critically, there is no crosstalk between the efferent and afferent signaling in the outer hair cells because there's no spillover of calcium, right? But can you comment just briefly on the cellular mechanism by which this compartmentalization occurs?
Yeah, this was work done by Marcelo Mogli and Juan Gutman. The base of the outer hair cells has an unusual high concentration of calcium buffers, oncomodulin and other calcium buffers. And also in the, at the base of the outer hair cells, there is a very special organelle, which is known as Subsynaptic Cistern, which lines all the base of the outer hair cells and in close apposition with where the efferent innervation takes place. So this sort of is like a physical barrier which prevents calcium spillover. So the connection probably of both of these mechanisms takes place to inhibit or to prevent spillover of calcium from one synapse to the other.
Great, thank you. And another question is the afferent neurons that innervate different regions of the cochlea, you told us show tonotopic specialization, especially type 1 neurons. Do the efferent neurons also show any tonotopic specialization based on which region of the ear they innervate?
They don't show really a clear, very fine tonotopic organization. We know in rodents that the highest degree of innervation is in the mid region of the cochlea, in the middle of the cochlea. And it winds down both at the apex and at the base. So it's not really a fine tuning of efferent neurons like we would expect from the fine tuning of the basilar membrane or of afferent neurons. But we only know that in rodents. We don't really know that in humans.
Thank you. Let's take I'm going to field the questions here just in the interest of time. A question from Jitu Mayor, who says he gave a wonderful talk, tour de force of molecular biology and physiology. He wants to know what the ligand for the alpha 9/10 receptors are in the context of the efferent system, and is this subject to central control?
The ligand is acetylcholine. It is a cholinergic synapse which releases acetylcholine, and that's the natural ligand. And the control, yes, it comes from the midbrain. It's a neuron that has its origin in the midbrain and goes all the way back to the cochlea.
And a question from Lou Sheffer. There's actually a lot of similar questions about the alpha 9, alpha 10 subunits, but is this type of, the extent of this diversity typical for all the receptors, do you think, or is this the acetylcholine receptor just an unusually diverse family? Because you showed that there are a lot of different possible subunits for these receptors. Is that typical?
So this receptor, the nicotinic receptors, form part of the pentameric family of ligand-gated ion channels, and that family includes serotonin type 3 receptor, and that serotonin type 3 receptors there are only 2 subunits, so that's very quite not that diverse. Then we have GABA A receptor subunits, and there are like 19 different subunits that can co-assemble, so that's quite diverse. And then we have the glycine receptors, and there are very few glycine receptor subunits, but I don't really remember how many. So it depends in which family of neurotransmitter receptors.
Sure. And do you think it's possible to trace the evolution of the alpha 9, alpha 10 channels to determine when in phylogeny this advanced function was actually acquired?
I mean, when did the alpha 9 and alpha 10 first appear? Was a sort of probably 10 years ago a paper by Dent where he looked for nicotinic receptors in non-vertebrate species, and he found them in non-vertebrate species. And originally it looks like there was an alpha 7 like subunit present in invertebrates, an alpha 9 like subunit, and then like a muscle, and then a neuronal. So it's very ancient. But exactly when it appeared, we don't know.
Thank you, Belén. I think in the interest of time, we should move on to our second speaker.